right, so the next panel is on fair and scalable data infrastructures. It's not easy to share large-scale imaging data. Some of them are terabytes, it's getting there. And then if you scan an entire human body, that becomes very quickly big data. And there's not just one human body that gets scanned, but hopefully many, so that it's equitable and not just male, but also male and female. And so very, very quickly across different ages, different ethnicities, different BMIs, it very quickly becomes large data. And it's not just imaging data, it's also single-cell RNA-seq and other data sets and many spatial data sets coming into existence. So we really need better ways of doing it. And let me see if I can promote others to come in here. So this is a panel which also has a lot of presenters, and we would like to have some discussion at the end. So it's great if you can keep it to 8 minutes. I know we have a lot of slides in the slide deck, and of course everybody wants to see you present, but it's also important to see us how we can have more synergies across these different efforts. And we already started to exchange data, exchange code. We learn together what works and what doesn't work for the single cell and other communities. So it's really, really nice to have this panel. And it is the last, but it's not the least panel. So my hope is that you will each share your slides yourself, but maybe I give a brief introduction to all of you. And again, this panel is on, let me just share my screen, is on fair and scalable infrastructure. We want both of this. We want that data to be easily available, but also to have large-scale data sets that are now shared. And Deanne is joining us. She is the Director of Bioinformatics in the Department of Biomedical and Health Informatics at the Children's Hospital of Philadelphia. Welcome, Deanne. Inno is joining us. He is at the Memorial Sloan Kettering Cancer Center. He is a bioinformatics engineer with an appetite for stats, visualization, pipelines, and DevOps. I like that very much on your LinkedIn site. Bruce Herr is the technical director here at CNS in the Intelligent Systems Engineering Department at Indiana University. Jonathan Silverstein is the Chief Research Informatics Officer at the Department of Biomedical Informatics. He is also critically important for getting everything in HubMap harmonized. We have 34 different assay types. He's also a visiting professor at the Biomedical Informatics Department at the University of Pittsburgh School of Medicine. And then there should be Maximilian Häusler joining us. He is a research scientist at the University of California in Santa Cruz. So welcome, everybody. And please, Deanne, if you are okay to share your own slides, that would be wonderful. And there will be a little timer in the top right, which Dan is running. I will ping Maximilian.
All right. So you can see my slides?
Yeah, not yet in presenter mode.
Yeah. There we go. All right, so I'm going to introduce this illustrious group of people with a little discussion on modeling human functional biology with knowledge graphs. And there's going to be several people that I can see from their slides that they'll be talking about similar things. So I'm going to be just briefly introducing my concerns. I'm also doing research through UPenn and CHOP on how are gene variations in disease and disorders related, because children often present first, right? When they're first born and there's something wrong, we usually try to understand what's going on with the kids and sometimes it's a genetic condition. So, you know, can we predict therapies given that particular gene variation? How can we help the children and their families? So my background is in physics and informatics. So my question is always, how do these data interconnect? How can we interconnect large biomedical data sets and use the structures and the information in those data sets to answer complex biological questions like diseases to genes, diseases to therapies, genes to therapies, etc. So knowledge graphs are essentially just In concept, they're simple. You're taking two entities that have some sort of relationship, either by knowledge or experience, that this disease does seem to respond to this drug, or this disease and this gene are related, and from animal models. So those kinds of relationships can be set up in different flavors, different types, and different amounts depending on the data set. And so node types would be like molecules, genes, or proteins. Those are the entities that we're connecting. And you can also have things like that connect them, like encodes or regulates or is associated or treats. And they're identical for connecting diverse data that would normally be more difficult to connect in a regular SQL database, or any other kind of way of representing that data, because you're actually free to consider how you would like to connect this data together in ways that might be meaningful for your biological or other type of question. It's even used by Google to drive their commerce, for example. There's a lot of ways that you can use knowledge graphs. In our case, we're using it to try to understand biology. So functional biology is essentially how genes and molecular processes drive development, functioning of cells, tissues, and organs on a day-to-day basis. A lot of that is done in the laboratory, trying to understand, if you tweak this, what happens to the cells or what happens to the human tissue if this gene has a variation. So knowledge graphs can actually integrate this functional biology information, and you can actually use it with anatomical maps, which we'll hear about later, and in concert with like the Human Reference Atlas that Bruce is going to be talking about later. This integration can give us tools to explain how genetic variations, for example, influence human development disorders and diseases, because you can map genetic variations, for example, onto the tissues they express in, or what kind of diseases affect what tissues and what parts of the body, and how do the genes turn on in those tissues. So I'll give you some examples, but first I want to talk about some knowledge graphs that we built, and Jonathan Silverstein has been involved with this with me. He's also on the call from HubMap. But the UBKG is a knowledge graph infrastructure that sets a bunch of interrelated concepts and medical vocabularies and ontologies. This is a really good way to understand how different ways of knowing things connect together. We, in concert with Jonathan, also started introducing genomics data into that network of knowledge. And we first built a smaller database called Petagraph, which has about 20 large multi-omics human and mouse genomic data sets. And it ended up with about 32 million nodes and 118 million relationships, some of that from the UMLS, which I don't specifically mention here, but that's one of the sources of the data. That's one of the sources of the data in the UKG. And then we extended that to something called the data distillery that was built in concert with about 11 to 12 common funded data coordination centers, including HubMap, GTEx, Kids First, 4D Nucleome, etc. And it's slightly larger, but it actually has a lot of data sets that are much larger and expertly curated and distilled down into the meaningful data content. So what we really want to get to is knowledge graphs that can enable a multidimensional human model, where you have both a map of the human body and the functional map of the human body, that you can actually bring in physiology, human genetics data, observational medical data, a dynamic biomedical process model, so that a doctor or a team can actually predict and treat complex disorders, and based on the patient's readouts or presentations, or even just a prediction of what their physiology should be like based on their particular set of gene variants. So an example of what we have here in the data distillery use case is we can look at fetal heart development by the developing mouse. So we can ask what genes linked to heart defects in mice are also actively expressed in developing human heart cells. So that might inform like what kind of genes could be, have variations or have mutations that actually increase heart defect risks in certain families. So here's an example, just a query for the data distillery. You can see the nodes there and their relationships. And we can ask what developing heart cell types to express genes also linked to heart defects in mice. And the query in the knowledge graph actually gives you a list really quickly, and takes seconds to complete, of all the different types of cardiac cell types. Whoops, pardon me for a minute. Oh boy.
We can cut that out, no problem.
There we go. Sorry. I don't know if my mouse sometimes does that. but the query will take seconds to complete, and you actually have the ontology or the identification of the cells, how the genes are expressed, and the particular symbols of the genes. In petagraph, there's a drug versus tissue effect use case that we can show where there was a particular drug that was recalled due to high cardiovascular risks. And the data was already integrated in petagraph. So we have rofecoxib, Vioxx gene expression profiles based on human that was put into Petri dishes of human cells and what kind of genes are turned on and off based on what the drug is doing in those cells. And then we integrate that with gene expression signatures across many human donated tissues. That was part of the GTEx project. And we can see really quickly in seconds what are the profiles of genes that actually look like look like the rofecoxib gene signature. And we found that according kind of matching what we already observed in humans, that there are basically the heart, kidney, and other types of arteries and heart tissues were also affected. So especially the heart was number one affected gene expression. So the query took seconds to complete. And this is the kind of thing you can do with knowledge graphs really quickly. You can imagine integrating all of this onto a map of the human body would be really powerful. These are fair and useful tools. You could download this with a UMLS license, and you can use a 16 gig memory laptop and install some of these knowledge graphs yourself. So it should be accessible around the world for anyone who has at least 16 gigs of memory. We suggest 16 gigs of memory, but people regularly use these graphs all the time at that level. And I'd like to thank the people that are involved with these projects. Thank you.
Thank you, Deanne. Great work. And I love to see these collaborations with HubMap and other efforts within the Common Fund data ecosystem. In the interest of time, it's my pleasure to give the word over to Inno de Bruijn. Please tell us more about H10 and the infrastructures that just got refunded there. So there will be a lot more coming. I will also post the link to the recent H10 package of papers into the chat for everybody to see. Thank you for running your slides, Inno.
Oh, I think you went mute. I missed a little bit of the last
Sorry.
bit of intro.
Thanks go to Deanne, and I love to see these collaborations across the different efforts, also within the Common Fund data ecosystem. And Inno is next, and I will post the link to the recent paper package that H10 put out in Nature into the chat. Thank you.
Thanks so much. Thanks so much for having me. And yeah, it's been really cool to see some of the talks and a really cool event. So yeah, thanks so much for including me here. I'm just going to talk a bit about the bioportal and the Human Tumor Atlas. So our team has developed two large portals for the sharing of multimodal cancer data. CBioportal on the left here has been in development for over 15 years and really had sort of this focus on bulk genomics data. And more recently, also more of a clinical focus, since a lot of these assays have been adopted in the clinic for cancer treatment. Then on the right, which more recent, so this over the last 5 years, we've been working on H10, the H10 portal which is for a consortium, the Human Tumor Atlas Network. And they use a wide variety of different assays, really focused on, single cell multiplex imaging, lots of the new data modalities that have come out. And yeah, so that's really on sort of the cutting edge of assays that are available. So yeah, just a bit of history on CBioportal. So this really started around the time of the Cancer Genome Atlas, which was really one of these first big efforts to do large-scale genomic sequencing across tumors and try to build a reference that we can look against. And so these were 10,000 tumors across different tumor types. And CBioportal really started as sort of an interface to sort of quickly query those results and look at them. And yeah, you see here over time how sort of the user visits have gone up, and it became sort of within the cancer research space really a very popular tool for looking at this data. And we started using it across multiple consortiums. We started using it at Memorial Sloan Kettering as really the place where people could go to look at all the clinical data that's produced at the hospital, all the genomic clinical sequencing data. And today we have about 100,000 or so patients in there that any researcher can look at and analyze. In 2015, we decided to go open source. It really became sort of a collaborative tool that we all worked on together. Different institutions joined. So although they really started at Memorial Sloan Kettering, Dana-Farber joined as well, and the Hive and other institutes. And actually one of the co-founders is Dana-Farber these days. So it really became sort of this multi-institutional and also multinational development effort. And so we've just been able to continue to grow. CHOP joined as well. We just saw the presentation from Deanne. So they're also part of the development team, and we continue to sort of grow it and trying to deal, with the huge amount of data that came in, lots of users that are visiting. So we got all these scaling issues that we've been trying to address in the last couple of years. And yeah, you can see, people from all over the world using it. We now have about 3,000 daily users or so, and all over the world, people also install CBio Portal themselves and load their own private data. So you can imagine different medical centers around the world that sort of have their own private data that they want to look at, and they sort of stick it into the stool. And we maintain a public version as well as several others for different consortiums. And yeah, you see H10 here also, where we sort of started to look at more how can we integrate these novel assays into that. So that's just a short history of CBio Portal. It's always been the goal really to make complex cancer genomic data accessible and interpretable for cancer biologists and clinicians. You can see here some of the different types of assays that it supports. So the scopinome data, expression, methylation, etc. And more recently, the imaging in single cell was trying to layer it in by sort of integrating with different other tools, and really with the goal to drive biological discovery and clinical interpretation as well. So those are sort of the two main use cases that we target. And there are a bunch of different visualization tools that we've built. So there's this dashboard kind of cohort exploration tool that allows you to see all the different cancer types in the cohort, their survival, different clinical attributes like smoking history. You can see the genomic changes in those cohorts. So you might know that TP53 is altered across a lot of different tumor types. So, about half of all the tumors will see that type of alteration. So you can quickly see that. And a nice thing with DNA is that, like it works across any cancer type, and really the data just by looking at the recurrence of something, it flows up. So you can immediately kind of spot it just by ordering the data, which is, much harder when analyzing imaging and other types of data. It allows different types of group comparison. So you can select any of these features, look at survival between those groups. You can look into specific gene queries. So you can zoom into a couple of them and kind of look at whether they're co-occurring. So we can see here IDH1 and TP53 mutations co-occurring. EGFR and IDH1 are mutual exclusive. You can also look inside the protein exactly how frequent a particular residue is altered. So you see this L858R residue that's frequently altered. And then lastly, there's this patient view. So you can see exactly where the samples were obtained at what time point in the patient's history, when surgeries were done, what kind of treatment they received, and then in each of those samples, what type of alteration they had. And we integrate with a lot of different resources that tell you about these alterations. So there's lots of alterations in a tumor, and some are driving the tumor, driving the growth, and we know you can treat them with particular treatments. So we have those kinds of resources in there, like OncoKB, that kind of give you that type of information. So that's just CBio Portal at a glance. You can go to the website to learn more about it. Then I want to talk a bit about the H10 portal. So this is really more of a data portal rather than an analysis portal. So really more focused on enabling people to get to the data, download it, and also explore it in different visualization tools. So we've added integration with different visualization tools to look at it. And see. So basically, H10 is really focused on transitions in cancer, trying to look at precancerous to malignant, and then eventually metastatic disease, as well as a response to treatment and treatment resistance. So it's looking at all these different transitions in cancers and look using a lot of different types of profiling methods to get an idea of what's happening in those different transitions. And most of these groups, they have a focus on single cell sequencing and multiplex imaging modalities. So here there's an example shown of a multiplex imaging of a liver metastatic biopsy of a breast cancer patient. And some of the things that it allows you to see, for instance, we can see here, the tumor has this high GATA3 expression. We can see the immune cells here around the border. We can see this immune sort of nest as well. So you can see these types of things. Perfect. So just want to thank the team, both the CBioPortal team and the H10 team, and I'll pass it on to the next speaker.
Thank you so very much. Great timing, great work. Super exciting to see that come together, and also to see the adoption rate over time, and that you now have so many users that really use that to make research decisions, but maybe also decisions related to patients. So next in line is Bruce Herr. I'm gonna run these slides here. I have them up. I have been trying to also get Maximilian. I'm not sure we are going to get him. But these slides are here, and I'm just going to put them in slideshow and share my screen, and then Bruce is just going to say, next slide, please.
My name's Bruce Herr. I'm the technical director of the Cyber Infrastructure for Network Science Center at Indiana University. And today I'll be talking about the Human Reference Atlas Knowledge Graph, and its infrastructure, its fair and scalable infrastructure of the HRA. So I believe it's showing my image now. So if you want to go to the next screen, as soon as showing the HRA, I'm sorry, I just can't see it, but I know what it's supposed to show. Is it showing the Human Reference Atlas now?
Yes. Mm-hmm.
Yeah. So I can do this. So the HRA, so what you see is, and you've probably seen this multiple times throughout the last 24 hours, is the HRA, and go next slide, you see where we have, we are breaking down the human into these anatomical structures, functional tissue units, cell types, biomarkers. We're trying to break down the knowledge. And if you go on the next slide, the HRA knowledge graph, we have a mixture of the red is like the knowledge that we're gathering from subject matter experts. They're providing it to us in various different formats, but to add that knowledge layer about the body down to the cellular level. And then we have in the green, the Atlas++. This is where we're adding in data from external resources, from experimental data, actual tissues extracted from real humans, running cell type annotation on those, finding literature about the knowledge in the graph, and then taking that further and bring them together. So how we then can, with the HRA knowledge graph, we can query across all these different layers to the ASCT plus B tables are the next slide. Do you hear me?
Yes,
yeah, okay.
we see the ASCT plus B tables.
All right, so the human curated ASCT plus B tables break down the anatomical structure, cell types, and biomarkers. That sort of is like the baseline layer of the knowledge, and a lot of things are just, you grab a kidney and you can find all the things connected to it. Okay, so next slide. Functional tissue units. These are these 2D functional tissue units illustrated and tagged with these ontology terms. So you can go up to the ASCT and Bs. Then we have the 3D reference organs, which are the same, only the 3D models that are also tagged, so you can find them. And they are, not only are these 3D models tagged, they're able to be registered too. So then in the next slide, you see where we have the experimental data from that Atlas++, where they've said specifically where they had extracted this human tissue that then we can associate the cells and the anatomical structures are colliding with the knowledge layer. So, and I want to try to get to the crunchy bits here, which is what's behind the scenes. So each of these boxes is, for the most part, they're all different types of digital objects, as we call them. A digital object has some data and has some metadata. And on our system, we take the digital object, and our digital object processor will it normalizes them to this LinkML form, which is a way to you can specify a structure that then has semantics associated with it. So we normalize the data to this LinkML form, which can then be readily translated into RDF and JSON. And so the digital object processor does that. So everything is in the same sort of form. So then each digital object becomes its own graph that can be queried by itself or combined on the fly to do different processing. And so all these graphs can then go into a triple store that then you can query on the fly using the HRA knowledge graph and using Sparkle, or by requesting the digital objects one by one. So then we build on top of that the HRA API and different applications that can then query that graph to add, programmer-friendly APIs or user-friendly applications to use this data and knowledge we're gathering. And so this is just the next slide is the ecosystem. It powers a lot of what we're doing with the HRA, and it is also how we're mirroring data to different portals like BioPortal and OLS and other soon-to-be other mirrors around the world. Okay, and then the HRA and KG data we have, next slide, has, so right now we have 10 million nodes, 171 million edges. It's about 125 gigs of the whole knowledge graph. And so this was released just Friday, actually. So the HRA version 2.2 has all this new data. And every 6 months, we refresh it with more data and more updates. And then next slide is just the growth per release. You're seeing we're adding new data types and just continuing to move it forward. And then usage is the next slide. And I'll try to work through this. And then the main thing I wanted to talk about is the way this is implemented is with linked open data. That's the next slide here. And I don't think I'm going to have time to go into this in detail, but from the beginning, with when we proposed what we were going to do with HubMap, we had this design for linked open data for our data. I think I'll just skip past the URL. So part of linked open data, if you look into it, like every thing has an ID, and you can look up that thing. So this Perl Human Atlas IO. A specific reference organ might have a specific ID that you can query. You can call out to that IRI. You can grab the data in the format that you want. Humans get HTML. A machine can ask for JSON or different formats. So the API is sort of built in for this semantic web sort of technology. And like one of the big wins with this, so if you go to the infrastructure, is the KG is pre-computed. It's a graph of graphs. It's all static data that we, when we compile it, it's static data that's delivered over the web and more or less can scale as infinitely because of the way it's structured. Because you have these graphs of graphs. You don't have to build the entire graph all at once. So we came up with a novel infrastructure. We're writing a paper about it. I would love to give more details on this, but I feel like I'm going to run out of time. So, but just want to point out that we've got this infrastructure is designed to be 5-star linked open data and can scale very, very much. And all of our data is CC by 4 and code is MIT licensed. And I think I'll leave it at that. sorry about my reception and stuff. It just is what it is.
Thank you, Bruce.
you.
Thank you for presenting. All right. In the interest of time, we do want some discussion at the end. Jonathan, are you ready? Can you share? That would be good. Thank you. Good to see you here.
So thank you for this great opportunity. It's really great to be at the end of this very long run. And I'll be talking on behalf of quite a large set of consortia on the topic at hand. So if I can advance these slides. Yes. The HubMap and SendNet programs and their fair and scale infrastructure is really what we're going to talk about here. So we have some presenters that have contributed to this, but this is what this team looks like. This is the SendNet team, and it's very highly overlapping with the HubMap data coordination team that is under multiple awards. SendNet is under a single large award. And so I've kind of combined this presentation to focus on different pieces of it, of these different consortia. So what are these? HubMap is, we've heard a lot about, I'm sure, and even in this last hour, we've heard about developing this open global platform to map healthy cells. The Human Reference Atlas is heavily related to HubMap. We have the marker paper, original one from there. Some others have been referenced. SendNet is a similar program that's really focused on, in addition to the infrastructure and data management and mapping efforts, it really has a scientific focus on the senescent cells. And there's a marker paper for that. At the bottom, I've described the general scale of these programs and their funding structures from the point of view of those worldwide that may be a little less familiar with the NIH Common Fund. So I have a number of slides here that are many for reference like this rather than going through them all. The HubMap technologies, I'm not sure if this slide has shown up here in these last 24 hours, but it shows the vast range of scales that these technologies cover. We've seen this from the point of view of the Human Reference Atlas, but the data sets themselves, as Katy mentioned, approaching a terabyte in individual data sets, but the data sets overall are hundreds of terabytes in each of these consortia across many, many different technologies and scales. So keeping those and managing them and organizing them over the long term, one really would like to have a large supporting computing infrastructure. And for that, we integrate very closely with and work very closely with Phil Blood at the Pittsburgh Supercomputing Center, where there are many, many years of this large-scale computing with national resources. This enables the ability to build the infrastructures that I'm going to describe on a way that we don't really, let's say, worry too much about scale, because as we need scale, we increase the scale. Far smaller, these projects, interestingly enough, are far smaller than many of the very large projects at the Pittsburgh Supercomputing Center, although much, much more deeply complicated. So you can think of these as being extremely wide data sets rather than tall, if you think about it from a simple sort of table format. In the advent of SendNet, which came a couple years after HubMap, we've moved and now been shown that we can build the same infrastructure on a completely different type of architecture rather than the large open computing. We've actually built SendNet onto a commercial Dell infrastructure at the University of Pittsburgh. And so we're running another local infrastructure for the other consortium. This is, I'm going to go to a quick look at a few things on the SendNet website just to get a perspective of the breadth of things that people do in a data coordinating center like this. This just shows the consortium undergraduate student program, which is a summer program for students coming and sharing within different labs. So we do everything from that to interview series of individuals to all of the data sharing and work on the underpinning data and so forth that we'll describe. But it's a complete set of infrastructure across human and computing. We do things one-off like the SynNet Biomarkers project. This was a project where individual folks came together to look at the literature and find biomarkers, and then have built this into a set of data that literally from a Google spreadsheet automatically generates this graph that people can explore and click on and find papers in PubMed. The main business here of this talk is this slide here, which really describes our flexible hybrid cloud microservices architecture. And this is really designed for every layer of data and software to be fair. I will describe what fairness is and this diagram in detail in just another moment or two. But the main points here are that we can run all sorts of different infrastructures on the same stack, whether in cloud or on-prem. So the blue things are the ones that are actually at the Pittsburgh Supercomputing Center, and the orange ones are actually in the AWS cloud. These repeatable systems allow us provenance, management of interoperability, and the open software data and publication policies with DOIs for every single component, both of the infrastructure and the data. So if we continue on along, we have the analysis pipelines that centrally process, which is a very relatively unique feature in this field, that each of these consortiums centrally process across all of these data and provide visualizations directly in the portal for each data set where that's applicable. The collection of metadata is very fundamental in working with a group at Stanford University with Mark Musin. We've used the Cedar Workbench and actually supported its modification to function not as pull-down lists and form filling, but as spreadsheets. So the entire thing can be pulled in as spreadsheets with their self-validatable referencing to ontologies, and really allow the metadata to be as fair as possible. The acronym for FAIR at the bottom there, I haven't heard it come up specifically yet in this talk, but I think this is the thing that we're pushing for: findable, accessible, and reusable. This is the example of 10x Multiome we heard from 10x in the last hour, and this is where you can download the template. You fill out the Excel template with detailed directory schemas, how you apply your data, and we ingest it. That very strict ingest path and pattern allows us to automatically process the data directly within the infrastructures over that large-scale infrastructure. This is a bit more from the CenNet portal now, that has a link out to show all of the complex flow of data from different labs to different types of technologies and different organs. And this particular one is live and interactive, where you can see into the machine that is publishing and processing all of these. So it doesn't just include the published data, but this gives you a sense of things that are in QA status and even that have failed out for a scale of what's going on behind the infrastructure in real time. These are some summaries just for reference here on the data in CenNet in particular. The data in HubMap is a couple years further ahead and about twice as much data. And then, of course, we maintain everything in order to generate fairness in the various repositories where they go. So this is protocols.io, which in CenNet has 160 different ones, and HubMap has many more. There's hundreds of there's about 100 repositories across the two projects in GitHub, 100 different ones. And then if we go further, let me just finish this up here, Katy. I heard the bell ring, but we've had the experience that others, because we published this DOIs and data site, and the others in the NIAID has actually ingested all of this data, much in a way similar to the HRA, on their own and created their own portal with sources of HubMap data. And we consider this a real demonstration of the fairness of this. So in summary, this is the main purpose here: the curation, ingestion, integration, findability, interoperability, analysis, sustainability. And then in this diagram, I'm just going to spend just a brief second on there. At the bottom, we have resources. Every single one of those services, ingest, pipelines, portal, all of it is working on this layered infrastructure through these APIs, so that nothing directly accesses the actual infrastructure at the bottom except through the APIs with the appropriate security. This has tremendous savings in cloud costs, having the infrastructure run the processing, maintain all the assets in the downloads, where the processing and the downloads are the most valuable to this kind of consortium, and yet they're the most expensive in the business model of public clouds today. So we can skip that, put our lightweight infrastructure in the cloud, heavyweight on-prem. And then I have some slides here demonstrating the portal. I don't need to go through those. Thank you.
Thank you, Jonathan. Great to have this presentation. And yes, we had a number of speakers point out that there will be foundation models, that there will be other AI models, and there will be a lot of compute power that will be needed to run this all. And so I wonder if your system architects have thought through how to support foundation models that are coupled, right? So that you can bridge those different scales. Your Levkovich presented a really interesting one. We had another team in China working on this coupling of foundation models also and presenting on it. And so I think you will have to provide a lot of compute power. And of course, you will also have generated data in addition to the experimental data. So how will you deal with that part? And maybe I just go around and start with Inno and then Deanne and then Jonathan again.
Sure. Yeah, it's a good question. I feel like for us, particularly I can speak on the CBio portal side, it allows for so many analytics in the web page. So we don't really, do much foundational model infrastructure support directly there. But you have thousands of users running all kinds of analytics. So you need to figure out how to support that. And I think there's a lot of, development in the space, how to make it faster, how to do it, but getting aware, becoming aware of that those technologies exist and how to best leverage them for your infrastructure is always quite complicated. Just to give an example, one of our databases, we've been using MySQL transactional database for many years, and then at some point somebody was like, hey, actually a lot of the work that we're doing is very analytical focused, so probably makes more sense to use an analytical database. So we switched to that, and that was a huge improvement that, something we just hadn't really looked at before, and just kind of came to be because somebody pointed it out who sort of came from a different, background. So I think that the interdisciplinary part is going to be very important, and people sort of helping you point things out.
Graphs are still kind of young in optimizing computation on them. One of the things that Jonathan's team does, and we've been trying to do with our queries, is trying to figure out, based on the structure of the graph and the structure of the data, what is the most efficient way to query it, because that makes a difference when you're doing production level stuff, like CBio portals doing, for example. We're not doing production level yet, so for us, we're in the research side, but still the data overhead could be quite large if you have very large graphs with a lot of calculations on top. So, part of it is software engineering, and part of it, of course, is the infrastructure. So right now, we're using on-prem computation. We don't do a lot of cloud stuff with this yet because we're not needing it. But when we do, we'll have to figure out we'll have to probably even get even tighter with our efficiencies.
Thank you. And you have the developmental aspect in there, right? So HubMap looks at healthy human, male, female, but you want to capture the developmental trajectories, and that's even more data. So looking forward to discuss that more.
Yeah, and there's the project, the DGTex project I'm part of, where we're doing the equivalent of GTex in human tissues, but we're going to be using spatial and single cell on healthy children's tissues. Mm-hmm. So there's going to be quite that 4-dimensional line now between, birth and even pre-birth, because we also have non-human primates. So calculating all of that is going to be another overhead, especially if and when we're probably going to be using graphs to manage that data.
I got Jonathan, the last word.
Well, I think on this question of scalability, we really focused on that from the very beginning. The National Science Foundation supports the expansion of what we're doing into free and open computing for anyone that can qualify for that. So on the analytics side, that architecture that we showed, in fact, the processing of the pipelines are in Docker containers that can chain from one to the other. So we've been able to bring in Azimuth, we've been able to bring in other computed processes. As you know, we're working on ones that have come from related Haggle competition, so they automatically process within there. And then I think in terms of the larger processing, one of the things that we've noted is that the limitations are more heading towards these large memory nodes and GPU nodes than the traditional scaled out computing nodes. And so fortunately, this is another area where PSC has been far ahead, and on the Dell infrastructures, we're adding more of these kinds of memory and GPU types of nodes to be able to handle these large data sets. We haven't yet, as a community, figured out how to do them in smaller memory. And this is where knowledge graphs are going as well. So I think one of the things that are extremely expensive are very high memory nodes in the cloud infrastructure. So once again, the on-prem infrastructure has some advantages there at the current time.
Use the best of both worlds, yeah.
Yeah. Yeah.
All right. I think that is the end of, well, almost 24 hours. I have a few parting words here, and my voice is getting crackled, apparently. It's a long sequence of events. So thank you all for joining us. I hope you can see my screen. We would love to thank all those that made this possible, so including CIFA also as a funder, but also the SAIS IT team here at Indiana University, which actually changed schedules around so that they could be with us over the weekend. We have Todd and Andreas, who compiled all the video collections that were running overnight. We have Melanie and Edward taking good care of the website. Nicole, Dan, and Meryl and Haley did a lot of project management and technical support. And Nicole and Meryl did all the social media notices. And so it was really, really neat to get this all come together. And again, it couldn't be possible without the community support. So we look forward to meet you in virtual environments or in person, ideally, at future events. See collaboration across these many teams. I think there are many, many opportunities to collaborate here. We are going to look out to watch the multiscale human data maps and models evolve rather quickly. I think with AI moving in, with all these new technologies that can generate tissue data faster and at higher quality and more coverage, it will be easier to make an atlas out of it. We will have to integrate data and code across consortia. So this last panel was especially near and dear to my heart. Thank you so much for coming in on a Sunday. And I think we also need more training materials on mapping and modeling the human body so that more people can contribute, and especially also ladies and gentlemen and others from countries such as Latin America and Africa can contribute. That was a topic in the last panel we had. We will add all the slides and recordings to the website. If you're really interested to see more and learn more about it, you might like to go back also into the previous 24-hour events. This is the fourth in the series, and I think we like it, but it is exciting and exhausting at the same time. But the slides and videos should be up mid-January. It always takes a little while to take out all the issues that arise over 24 hours. Here are some of the events that you might like to join us for. This is a more industry-focused event, but there is a track on knowledge graphs, so hopefully I see some of you there. There's also the Junior Investigator Meeting in 2025. This event has been great in bringing together the next generation of Atlas makers. So here we know that it will be in the DC area. Dates are not set yet, but last time we had 140 young investigators come together, many of them in person. And it's just wonderful to see all that innovation happening in this environment. There is a save the date also for the next general meeting of the Human Cell Atlas in Singapore. So hope to see many of you there. I know it's half around the globe, but still I think it is important to go to Asia, to go to Africa, to go to Latin America, to bring in those researchers that we absolutely want to have contribute to this so that it's a truly equitable effort, that not just the data comes from all the different ethnicities we have here on this planet, but also the researchers come from those ethnicities, and it takes extra effort to empower them. There's also a systems biology of human disease event. So again, save the date. It's in June. It's in Berlin, which is a wonderful city in the middle of Europe. And please do come join us. Yeah. Last but not least, I wanted to let you know that our team recently committed to another 10 years of the exhibit, the Places and Spaces exhibit, which we have been running for 20 years now. This third decade will be on envisioning intelligences. We believe we have to visualize how intelligences operate, how they collaborate, compete, and ultimately succeed together. And so we will include all kinds of different types of intelligence, from linguistic to kinesthetic to communication, musical, emotional, and other intelligences, with a focus on collaboration and coordination across life forms. And I think that makes it especially interesting because it's not just human and machine, it's also fungi and trees and human and microbiome and many other collaborations that are absolutely needed for us to digest our breakfast or soon lunch here. So we absolutely need to understand that better. We need to communicate it better to general audiences. And we hope to also inspire discussions about how these intelligences operate, how they should operate, how we want them to operate in desirable futures. So I think with AI moving in, this is especially interesting. And there might even be a 24-hour event next year on collective intelligences and what we can do together in the biomedical domain, but also in arts and humanities. So please stay tuned and come back if you can. And happy holidays and a wonderful start into 2025. Thank you so much for joining us all. And I think that is the event of 24 hours. Thank you, Dan, for also the technical support here. And Bruce and everybody here on this last but not least panel. Great to see you all.